Hello, everyone. Welcome back to our channel. Today, we have an important topic to discuss: how to register your isolate genome project that you sequenced in the lab but have not submitted to public repositories such as NCBI, along with its information into the Genome's online database, also known as GOLD. First, let's start by opening our web browser and navigating to the GOLD homepage. Once there, you'll see a register button. Click on it to begin the registration process. Now you need to specify the type of project you want to register. In this example, we'll start by creating the sequencing project. Click on the arrow next to sequencing project to reveal more options. Since this is not a public NCBI genome, select that option. Please note that users do not need to manually submit public genomes from NCBI. If you have one to add, all you have to do is go to our help section, send us a quick note, and we will import it for you. Coming back to our genome, once you have clicked on the sequencing project, you have the option to search for your study name if you want to use your existing study. If there's no match for your study, click the button to create a new study. Provide a short description and fill in the ecosystem information. If you have additional information, you can include that as well. You will find two options to select from here: biome and organism. If yours is an isolate genome project, you should choose organism. On the other hand, for metagenome and metatranscriptome projects, you'll need to select biome. Since we are working on an isolate genome project, please click on the organism option and proceed to create your organism. Please perform a search to ensure that your organism is not already present in the GOLD database. If it doesn't exist, you'll have to create a new organism. You have the choice between cultured and uncultured organisms. In this case, my organism is cultured, and it was isolated by either me or by my colleague. Note that if your organism was isolated from a host-associated, soil, aquatic, or any other environment listed below, you have an option to select the appropriate package and get access to the form with additional metadata fields specific to the respective environment. Otherwise, choose the standard package to access the default organism entry form. For our example here, I will be selecting the standard package. To save time, I won't go into detail about all the fields, but make sure to complete the necessary information marked with an asterisk. If you have any uncertainties, don't hesitate to hover over the Information tab for more assistance. Finally, we'll create a sequencing project. Fill in the required fields just as we did with the organism registration. Make sure to provide accurate information for your project.
Now we can make our analysis project. Most of the information will be autofilled, so we can put in the assembly method and press submit.
That's it. Congratulations, you've successfully created your isolate genome project in the Genome's online database.
If you wish to submit your data to IMG, you can do so now.
If you have any additional questions or would like more information, feel free to check out our help section. In our help section, we provide detailed instructions on how to create different types of projects in GOLD. Here, you will also have access to commonly used GOLD terms, an elaborate FAQ section, and much, much more. Thank you for watching, and I hope this tutorial has been helpful. If you have any questions or need further assistance, please leave them in the comments below. Don't forget to like and subscribe for more informative content. Until next time, take care.